DNA replication is the process of how DNA makes a copy of itself. It's essential for a cell, or else it wouldn't be able to grow through mitosis or reproduce through meiosis. The process of DNA replication first begins with a complete parental double helix strand right here, and the enzyme helicase begins the process by unwinding the base pairs bonded with H bonds, and it creates this replication fork where the top and bottom strands separate. And these single-strand binding proteins bind to the newly single-stranded DNA near the replication fork to stabilize DNA before DNA polymerase comes in, reads and binds the appropriate base pairs to create another double helix. So the leading strand works from the 3' to the 5', or the 5' to the 3' going this way towards the replication fork, while the lagging strand, because it codes from the 5 to the 3 in this direction, it creates these Ogasaki fragments, and this is where DNA ligase works to finally bind them together. Over time, cells get damaged and need repairs to keep them from dying. The mechanics that work to repair cells and mutations that occur are various enzymes. For example, sunlight exposure can damage skin cells, and an enzyme called nuclease works to cut out and repair the damaged DNA.
Now we will talk about transcription. The purpose of transcription is to convert DNA into messenger RNA, and this happens in the nucleus. So it begins with the tau box, which is in the promoting region of the gene, and it just shows where the gene begins, so this being the rest of the gene. And these are transcription factors, and their job is to act as launching pads for the RNA polymerase, and they also show it's also how the RNA polymerase finds the gene. As the RNA polymerase moves down the DNA, it unwinds it, and it copies the information from the DNA by making a complementary strand of RNA. So in the end, this strand of RNA has all the same information from this original set of DNA. So after termination, the DNA will zip back together again by hydrogen bonds, and we're left with this RNA, and then we will put a 5' cap on one end to signify the beginning of the strand for translation, and it's also for protection, and then on the other end we will add a poly A tail, which is just a bunch of adenines, for protection. So within the RNA, there are introns and extrons, where the introns actually just contain unnecessary information. So what the spliceosome does is it snips the RNA, removes the extrons and splices together, removes the introns and splices together the extrons, so then you're left with mRNA. Within the spliceosome, there are protein structures and RNA complexes. And so this snRNA is made up of ribosomes, and it acts as a train track for the RNA. And then the snRNP acts as structure for the spliceosome, and it's a protein. Next, we move on to translation, which happens in the cytoplasm. The purpose of translation is to create proteins from mRNA, which was made in transcription. So in general, what happens is as the mRNA moves through a ribosome, polypeptide chain is made, which eventually becomes a protein. Each ribosome has a small ribosomal unit and a large one, and in the large one there is 3 binding sites, E, P, and A. Within mRNA, every 3 bases makes a codon which matches with a specific amino acid. So for example, AUG matches with methionine, and so once the mRNA reaches the ribosome, it awaits a transfer RNA to bring it an amino acid. So the purpose of transfer RNA or tRNA is to bring amino acids from outside, from the cytoplasm back to the ribosome, so that it can add to the polypeptide chain, which we saw earlier. There are 3 stages in translation, initiation, elongation, and termination. So in initiation, a tRNA binds to the P site with the codon AUG, and with, like I said earlier, with amino acid methionine. And on one end of the tRNA is methionine, the other end is an anticodon, which bonds with the codon. Once the amino acid is attached to the ribosome, this transfer RNA can leave its amino acid, move to the A site, and then the tRNA that matches with the next codon can fill the E site. During elongation, the polypeptide chain grows as each codon matches with the transfer RNA that brings an amino acid. So this will keep on growing as this goes through the ribosome, and these keep on doing their job. Lastly, termination occurs when a ribosome runs into one of three codons. So, for example, this codon UAA is a stop codon, which doesn't match with an amino acid, but instead just tells the ribosome to stop the process of translation.
Problem that can occur in DNA replication is if a change to this nucleotide sequence occurs, and whether it's substituted, it's added, and this whole thing is shifted down, and then it becomes a code. They code for different codons that may not fit together, and these codons may not code for the protein that is needed. Right here is an original nucleotide sequence, and so normally they code by 3. Every 3 nucleotides codes for a specific codon, which then when they're all added up, they create a protein. Problem that can occur in DNA replication is if a change to this nucleotide sequence occurs, and whether it's substituted, it's added, and this whole thing is shifted down, and then it becomes a code. They code for different codons that may not fit together, and these codons may not code for the protein that is needed.